Continuing on, our next presentation for this morning's session is entitled Genotypic Characterization of the U.S. Peanut Core Collection, and our speaker is Paul Otyama.
Thank you for the kind introduction. Let me share my screen here and hope you can all see it. So good morning. My name is Paul Otyama. I'm a PhD student here at Iowa State University. The title of my talk is Genotypic Characteristics of the U.S. Peanut Core Collection. Now in this talk, I'll be talking a lot about germplasm collections, and I want to introduce that. And then after honing on the U.S. Peanut Core Collection and explain just why we wanted to genotype this collection. And then in the last segments of this talk, I'll present results from genetic structure and genetic diversity in this collection. Now about germplasm collections, these can be really large, and the fact that they're very large limits their usability and characterization. Now in 1984, Frankel and Brown introduced the concept of core collections, and following that, in 1993, the very first peanut core collection was created by Dr. Holbrook and his colleagues. This collection has 831 accessions, and the total germplasm collection in the USDA category has over 9,000 accessions. In 2000, another core collection was released, and this was the APRES peanut core collection. This is a much larger collection with over 1,700 accessions. Shortly after that, the very first of the mini cores was released, and this was again by APRES, and this mini core collection has 184 accessions. In 2005, Dr. Holbrook again and his colleagues released the US Peanut Mini Core Collection, and this has just about 104 accessions. Now, much recently, the Chinese Core and Mini Core collections were also released with 576 accessions and about 300 in the mini core. Now, germplasm resources are really great sources of important alleles that we can introduce into peanut cultivars. And that's really one of the reasons why we thought to genotype this collection, the US Peanut Core Collection. Now, I want to mention here that this data is available and you can access it through the AgData Commons or the peanut-based data store. The reason why we genotype this data, and as you can see here in this schematic, that figure that I'm showing here, all the accessions in the US Peanut Core Collection come from all over the world. So it's really pretty diverse in terms of country of origin. And we wanted to attach genotypic data to each of these accessions and make this available. Now, I want to talk a little bit about what data is available. I mean, given that this is a grad student presentation, you never know, this could turn into a master's thesis project for you, or maybe a PhD chapter for your dissertation. So we have the original SNP calls from Affymatrix, and these are really the high confidence SNP calls that are polymorphic in this collection. We also did some downstream filtering and we made this available in a VCF format. Now, beyond that, we wanted to check and see if we have any variants in the sequence genomes that we have. We have 3 sequence tetraploid genomes, Tifrana, Shitoki, and Fuhuashing. And then we created sort of like synthetic tetraploid out of the progenitor sequences of Arachis and APRES that were available much earlier. So with these, we called SNPs from the positions that we already have in the Affy data, and we make this data available in 2 formats, a tab-separated format and a FASTA format. In addition to that, we took 16 highly diverse accessions from the core collection, and from this, we wanted to check the origin of each of these SNPs. If we could be able to infer subgenome origins for these SNPs, and the value of this I'll present in the later points of this talk. But this data is also available, and you can see that I've just mentioned a little bit of the data, but there's a lot of it, and you can make great use of it. So I encourage you to do that. So let me jump into my bias, the reason why I wanted to sort of study and answer questions using this collection. Back in 2019, we did some analysis with the peanut mini core collection, and from it, we found that there was a high rate of genetic similarity among accessions. Now, even if the mini core has about 112 accessions, when you're accounting for this high genetic similarity, you find that the overall effective collection size is much smaller than 112. We found that, this genetic similarity was sort of structured. So within the whole collection, you have different stratas of groups that are very similar and quite different from each other. And we thought this possibly extended to the overall germplasm collection. They didn't have a way of answering this question except by genotyping the larger core collection. Some of the questions that we're interested in answering that we're not able to elucidate on using the mini core was, what is the nature of this structure that I've been talking about? Can we explain this structure using any biology that we know? And beyond that, how much genetic diversity do we actually have in the core collection if, like I said, we are finding high genetic similarity? So we went ahead and we did a phylogenetic analysis using the sequences from the core collection. And this is the phylogenetic tree that I'm showing here. What we found was that genetic diversity in the collection could be summarized in 4 main clades. Clade 1, 2, and 3 having upwards of 200 accessions, and then clade 4 being a much smaller accession. Now we couldn't place a transition clade here, clade 3.2, and we just included it there. But this has this has way fewer accessions in it. So even if this collection is really large, you can see that we have about 4 main groups in this collection, and it's not homogeneous at all. Another finding that we had are these that I'm going to be pointing out. So when we got the sequences from the genomes that have been sequenced, Tifana, Shitoki, for washing, and then the synthetic tetraploid that we created, we found that Tifana clustered in clade 1, and the 2 Chinese genome clustered in clade 2. Right away from this, you can make a conclusion that although the 2 Chinese genomes are very similar, they're quite different from the Tifana genome. The other important finding that we found was that this synthetic tetraploid that we created clustered in clade 4. Now, clade 4 is pretty interesting because this clade, with just 84 accessions, was mostly predominated by accessions from Bolivia. All over the tree, there is a high presence of accessions from South America, but in all these clades, there are interspersals of accessions from all over the world. And that's a significant finding because we know that peanut had mainly diversified into modern cultivars before it was dispersed all over the world via trade routes. But then finding the tetraploid sequence in a Bolivia-dominated clade was able to sort of throw some more evidence into the origin of peanut. And we believe that to be around southeastern Bolivia or northwestern Argentina. Now, beyond that, I wanted to see if we could visualize this structure in a different way and hopefully outlay biology onto it. So we took a set of unlinked SNP markers and we did a principal component analysis. And what I show here on the x-axis is a plot of PC1 by PC2. And together, these principal components explained about 60% of the variation in the data. Now, the data points are just sequences from each of the accessions in the core collection. And, right away from this, you could see that the diversity is, quite spread out. But beyond that, I wanted to see if we could make some more information out of it. So I'm going to paint each of these accessions with a color according to where they cluster on the phylogenetic tree that I showed you much earlier. And when you do that, you can see here that the clade 1s, sort of cluster neatly together, as do the clade 3s, you find that accessions in clade 4 cluster almost central in this diversity landscape that I'm showing you. Conclusion from this is that the phylogenetic tree truly captured the structure in the core collection. So I want to overlay information onto this clade data and see if we can make some inferences in biology. And the first that we did here was botanical variety. And you could see here that the botanical variety also clusters quite neatly following our clade analysis. You see here, down here, that most of the accessions that are green are fastigiata, and then you have the vulgaris varieties, clustering mostly in clade 2. We did the same with market type, and you can see all the Valencias, almost falling in clade 3, and then you have all the Virginias falling in clade 1. Now, these two categories that I described, market type and botanical variety, are important in peanut classification because they tell us a little bit about subspeciation. Next we wanted to check, I talked about the minicore earlier and the predictive analysis with it. Well, we wanted to see how well the minicore represented the diversity in the core collection. So we overlaid this information on this graph here, and the red represents the minicore accessions, and then the sort of blue represents the core accessions, and then you have cultivated in them. The observation in this, you can see that the minicore accessions, pretty much span the entire diversity of the core collection. So given that, this collection was created just based on phenotypic information, I would say Dr. Colley Holbrook did a really great job in creating a nice collection for the community to use. So I'd say well done and good job, Dr. Colley. So the main takeaway from this, we found that subspecies and botanical variety classifications are really important correlates of genetic diversity in the core collection. So beyond that, we wanted to survey the status and the nature of genetic diversity in the collection. And what we did, and there were findings that peanut has very little genetic diversity. And this was mainly because tetraploidization of peanut is fairly recent, and this was out of a severe genetic bottleneck, so there might not have been a lot of time for genetic diversity to accumulate. So in subgenome exchanges, we find significant sources of diversity in tetraploid peanut, and wanted to see if we could show this. So what I'm showing here is all the 20 chromosomes of peanut divided into the A subgenome and the B subgenome. And in all the places where you see the red here, these are places where we're able to identify that these DNA variants are actually coming from the opposite genome. So the red that you're seeing here is coming from the B genome, and the red that you're seeing here is coming from the A subgenome. From this, you're able to conclude that there's a significant amount of subgenome exchange that is taking place. And this is about 10% of the alleles have arisen out of subgenome exchanges in the peanut genome. This is widespread, and it can be at a chromosomal level as well. So the main conclusion from this study was that diversity can be summarized by 4 main clades, and that structure, correlates well with subspecies and botanical variety. And many of the alleles in the peanut genome arose out of subgenome exchanges. With that, I'd like to acknowledge all the co-authors that worked on this paper, especially Dr. Roshan Kulkarni, who's soon to be a doctor, and then our great collaborators from the University of Georgia, the University of Oklahoma, and all the people that make it possible for us folks down here in Iowa to do peanut research. This work was funded by USDA and NIFA, the National Peanut Board, and the Agriculture, Food and Research Initiative. With that, I'll take questions. Thank you.
Thank you. We have time for a couple questions and a very interesting presentation. The first question comes from Shyam Tullery, and the question is, based on your findings, would you be able to suggest what % or how many of the 831 core collection genotypes could be truly unique? So based on your findings, would you suggest what % or how many of the 831 core collection genotypes could be truly unique?
That's a great question. Based on my finding, how many of the accession and the core are truly unique? I would say, you know, and being very conservative, don't quote me on this, it could be about half of them, you know, just being unique, and that's about just 400 of the accessions. So there's a lot of similarity among these accessions, and I'll throw a caveat in that the SNP positions that we have are only 13,000. So we're not really fully surveying the whole genome for us to be able to make conclusions about similarity, but there's a high degree of it. Yeah, that's a great question.
Okay. That concludes the time we have for questions, but Dr. Charles Chin at Auburn University has a question for you as well, so you might follow up with him. Okay.
Okay, thank you very much.
Thank you very much.